This is Digital Pathology Today. Now, here's your host, Dr. Joseph Anderson.
The big trend in oncology over the past few years has been immunotherapy. This trend is converging with the digital transformation we are undergoing in pathology. We seem to be at the crossroads of immuno-oncology, digital pathology, image analysis, artificial intelligence, and the increasing ability to multiplex. Welcome to Digital Pathology. I'm Joe Anderson. Our guest is Dr. Keith Wharton from Ultivue, a company looking to reveal actionable biology through multiplex immunofluorescence to make immunotherapy a reality for patients with cancer. Keith is a board-certified anatomic pathologist with diverse achievements in research, drug and diagnostic development, as well as clinical investigation. He leads Ultivue's pathology and biomarker analytics team. We're going to be talking about what exactly is the need for multiplexing, how much information can multiplexing add above and beyond singleplexing or other histologic features? What about diminishing returns, and what are challenges to implementation? And what about the increased system complexity that comes with multiplexing? What about system failure? And we're also going to look at specific applications in immune-oncology, multiplexing versus single gene analysis of markers such as PD-L1 by other methods such as IHC, or histologic features such as tumor-infiltrating lymphocytes or TILs. Can these be used alone or in a complementary approach? Dr. Keith Wharton from Ultivue, welcome to the podcast.
Thank you.
Yeah, this is very exciting times. So I think a lot of folks are interested in what you're doing at Ultivue and multiplexing and digital pathology. So let's talk a little bit about that. I think immune-oncology, also is a very hot area, and it sounds like you have some applications in that area and probably goes beyond that. So let's just jump right in and talk about what is ISP technology.
Thanks, Joe. ISP stands for in situ plex. So this is a DNA barcode-based antibody staining and signal amplification system for multiplex staining. So it was developed by Ultivue several years ago now. Ultivue, by the way, is a spin-out startup company from Harvard's Wyss Institute, who was founded by David Walt and Peng Yin, who are faculty at Harvard. It's about 6 years old. We've been through 4 funding rounds. And so back to ISP, 4 basic steps. It's a more or less standard staining workflow. So you take a unstained section that's been properly prepared in a standard pathology lab. You apply a set of DNA barcoded antibodies. That's step 1. Step 2 would then be doing in situ DNA amplification of the barcode on the antibody using a proprietary technology developed in-house. The next step then is, since you've amplified the barcode, now you hybridize fluorophore linked to a complementary barcode. And that creates a bit of a Christmas ornament type arrangement, and then that amplifies the signal. Then you can detect using a variety of fluorescent slide scanners, or even using an old-fashioned fluorescent microscope, although most people don't do that these days.
Okay. So it sounds like it's DNA-based. It sounds
Yes.
like there's an amplification step, there's a hybridization step, there's a fluorescence-based detection. So it sounds like there's a lot of similarities to FISH, but I'm guessing it's kind of a proprietary technology, so it's not exactly the same.
Some similarities to FISH. The big difference is that the antigen retrieval is pretty gentle, right? In this case, the DNA to be amplified is kind of sticking out of the antibody instead of buried somewhere in the cell or in the DNA. So the antigen retrieval standard, it's very gentle in the tissue, and the staining could be done on automated immunostators. And right now we have predominantly used the Bond, Leica Biosystems Bond RX. Another nice thing about the gentle aspect of the tissue is that you can take off the probes through an exchange process, dehybridizing the probes, and then do another round of staining.
I see.
So you can get up to 8-plex, 12-plex. We've done 16. I think we've done 20 in our research group. So possible to go higher. Of course, it's more time, but it works pretty well. 2 other things, if I can mention them, is that we have the ability to stack those MIF, multiplex immunofluorescence data sets to get the 12-plex, and then, of course, you can false color to visualize it any way you want. But another thing we can do is merge an H&E of exactly the same slide that's been imaged, and then we can merge that with the multiplex fluorescence image stack using a software called Ultistacker that we've developed in-house. So this lets you phenotype, in essence, every cell in the section. I think this is an incredibly powerful technique to visualize the marker phenotypes in the same section where you see the H&E.
Right. So you're able to overlay those images really to get
Right.
a very precise immunophenotype, and you can localize it to the H&E features.
Right.
And the multiplexing, I think, is a giant step forward, right? Moving away from, one or two markers to being able to multiplex, I think is very intriguing. So interestingly, you use fluorescence, and I think FISH, how we used to utilize FISH, I think one of the main applications was looking at HER2. And I think that market kind of moved away towards CISH, right? A more chromogenic based detection system. So, and then you mentioned you were utilizing the Leica Bond instrument. So I'm wondering, what are there advantages to fluorescence-based detection system compared to a chromogen? And, how did you go about evaluating that? And, is it possible you could move to a chromogenic system in the future?
I guess we could get into the differences between bright field and fluorescent microscopy. I think they're important. Of course, pathologists have a lot more familiarity and affinity for bright field microscopy. So that's one big driver of getting all assays in a bright field format. I frankly think that's going to matter less once there's a much wider adoption of digital pathology. And the output of any assay that's done on the tissue can be colored or visualized in a way that is, like a bright field microscope. So whether it's a pseudo-color overlay or a symbol denoting a particular cell type, I think the sooner we're able to get the data off the slide, whatever the modality is, into a data format that's compatible with digital pathology, I think the modality will matter less and the quality of the data will matter more. And I think there's a lot of literature about the advantages of fluorescence detection as a means to localize proteins, to understand the dynamic range of expression, to look at co-expression. I think those technologies are, at least based on current comparisons, are far superior to chromogenic. But you are right. Pathologists have used chromogenic IHC for decades, are very used to it. They're used to doing one stain at a time and signing out cases that way. So I guess I feel like digital pathology itself is foundational for formatting data in a way that it really doesn't matter where the data comes from or how it gets there, as long as it's trusted.
Yeah,
by
that's
the
a good
pathologist.
point. Yeah. I think as we increasingly go digital, maybe the pathologist's familiarity with these things is going to matter less. And, we go from directly to the scanner and then visualizing it on a monitor or computer display. I think those considerations may become less important. And interestingly, so it sounds like there's clear advantages to fluorescence. Maybe the signal is crisper. You have a variety of different colors, and maybe it's easier to discriminate either by, machine learning or image analysis.
Right. Well, once again, there are colors that the fluorescence uses for detection, with the filters and the camera and so on. And then you can color, in terms of visualization, you can color the false color the images any way you want, right? Depending on how you want to visualize them. So I think there's two ways to think about color there. One is the technology. The other is how you use it to aid interpretation.
Yeah. And so what about multiplexing? I think it's very intriguing. And, I think we've all been kind of waiting for this moment where, A, we're going to go digital, and B, we're going to be able to multiplex to our heart's content. Right. And so, but I think there's a downside to it, or there's technical considerations, and I think, maybe starting at the top, genes often cluster, right, in pathways and families. And I think there is maybe a natural tendency to think, well, more is better. If we can have 10 genes, why not 20? And if we have 20, why not 100? Yeah. That's got to add more information. But the downside is maybe it adds complexity. And maybe there is diminishing returns in terms of, the information you can get about the biology or the pathways involved. So what's been your end? And I think failure, right? Like, is it, more likely to fail? Let's say you have a 10-plex assay and 9 out of the 10 work. Maybe you'll have to throw it out, right? You're going to need all 10 to work every time. So I think, are there considerations in terms of finding that sweet spot as to what is the best number of genes to look at?
Well, first of all, I think in terms of the number to look at, the nice thing about multiplexing is that I think it fits in sort of a middle ground between, one marker per slide per IHC, which lets you answer a certain set of questions, and the sort of single cell omics, spatial transcriptomics techniques that let you answer, very deep questions and actually perform discovery, but I think are probably not scalable for the use of a typical pathologist, or they won't care about a lot of the data. It'll be a bit like NGS, where if you do a large panel, a lot of what you get is not useful, but maybe a little bit of what you get is useful. So about diminishing returns, I think what you mean by that question is whether you get less extra value from the information you get as you add each additional marker. And I guess I feel like one advantage of multiplex panels is that you can choose a set of markers to answer a very specific set of questions you have. And again, whether it's research discovery or diagnosis. So I think if you're designing and using the panels properly, there shouldn't be any waste, because the markers you pick, the channels you use, the analysis you do are designed at answering the diagnostic questions that you want to answer. So that's for diagnostic use, but then for research use, you do want a lot of extra data, because you don't know necessarily what you're going to find or what you're going to learn. So it depends on the user, I think. System complexity, I think you touched on. That's a bit of a tricky one, because of course, as you know, the only prevailing digital pathology systems that have been cleared by the FDA are really only to be used in comparison with a microscope, to be viewed by a pathologist without image analysis, without AI, without anything. So it's just, are they comparable to a microscope? So then when you add multiplexing on top of that, right, you need probably a different scanner. You probably need more image analysis for sure. To make sense of the image analysis, you probably need some sort of a reference atlas. And then to really kind of understand it or make sense of it, you'll need probably deep learning or AI of some sort. So, this ideal system of the future we can speculate on later on. I think we're going to get there probably sort of in a stepwise fashion. And again, I think the first step is just getting pathologists comfortable with images on a screen and looking at the data and comparing it to what they're used to, which is DAB, IHC. And I guess one last point, you say, so let's say you do a 10-plex and then one of them doesn't work. I think it depends on the use of the 10-plex. Like if you just compare that straight up with 10 IHCs, you could argue that most of the IHC done today is, redundant. It's part of panels. It's low risk class. You rarely make a decision on one slide, the information from one slide, or the success or failure of one marker, right? You have internal and external controls. I think it's possible for any one marker to fail for sure, but I think if you've designed the experiment correctly, you'll make it robust to failure of any one marker with the proper lab controls and proper perhaps on-site controls. So again, a lot of topics there. I hope that got to most of them.
Yeah, absolutely. Yeah. Sorry, I just kind of got so excited, and a rush of questions came into my head, and I'm also showing my bias. I'm looking, from the perspective of commercially available molecular-based assays. So yeah, I guess my point about diminishing returns is, I've seen personally and just through study molecular assays on the market, that may have 10 genes, 20 genes, and so on, and they come up with a composite score.
Yes.
But much, I think in one of the cases, a well-known assay, over 50% of the information could be derived from one gene, right? And then,
I see what you're saying.
And then so as you add more and more genes, I think progressively they add less and less value. So then, it's almost an arbitrary decision where you have to kind of make a judgment call. Where do we cut this thing off? I think it's, we want it to be as informative as possible, but we want it to be as simple and robust as possible too.
I guess I'd have to believe it's a bit of a different game. The assays you're talking about, right, are based on bulk transcriptomics, where you really, if the cell's important, pathogenic, but if it's at low abundance, right, the signal from that marker is going to be diluted in the bulk transcriptomics. I think the single cell analysis as a foundation of these panels is going to change the game, because the pathologist already has the ability to look at every cell in the biopsy, right? That's their job. So to have that information overlaid on every cell in the biopsy, it's a different set of questions and challenges you come across. And the one that bulk transcriptomics faces with, should it be a 12-gene panel or a 15-gene panel? It's a different problem, I think. In fact, that other problem kind of goes away.
I see.
Frankly.
Yeah. And then so kind of along those lines or along that bias is, I'm thinking of it maybe more from the perspective of a commercially available assay or an assay that's designed to be used clinically. And
Yes.
then you have other considerations, like mainly that it needs to be robust, meaning that it has to work most of the time. Otherwise, you can have the best assay in the world, But if it, even if it works even below 90% of the time, it's going to become very frustrating, right? You have doctors
Right.
and patients counting on these results, and
Right.
oh, lo and behold, sorry, you're the 1 out of 10 person we can't get results on,
Yeah.
and then it has to be able to be done relatively quickly with a fast turnaround time. But you bring up a good point that you have to be clear about what you're using it for, and there are uses beyond clinical use, mainly research and discovery. So
Well,
maybe talk. Go ahead.
No, I was going to say regarding robustness, I think, right, you can test that. And I think there have been a few publications recently using our technology and others about reproducibility. I mean, there's no doubt to do multiplex fluorescence well, it's more effort than doing a manual H&E, let's say, right? There's all the usual upstream pre-analytic staining considerations that need to be paid attention to, quality of histology, slide quality. Then you have to have robust reagents. They have to perform on a robust platform. And then, you need a scanner that gives you an image that is, reflects what's on the slide, right? There's more steps, but I think that's testable. Of course, if any lab decides to develop a test based on this workflow, right, they have to define their system and validate it anyways. And so I think the drivers there are, at least initially, in the initial stages of adoption, assuming the other aspects are green lights, are just redundancy, right? Pathologists like to see, the DAB bright field IHC as a comparison to make sure that what they're seeing, they can trust. And that's in fact, how we QC our assays when we create them and we make kits. So redundancy is, in design and in testing is helpful for robustness, beyond the actual design of the assay. And I think with turnaround time, which you mentioned, clinical workflow turnaround time, I think that has to do with how well it fits into an existing workflow. So, this is where I think some of the technologies, either from an assay development standpoint or assay turnaround standpoint, are very challenging to implement clinically unless you're just willing to wait for 2 to 4 weeks to get your results. And I think nobody's going to be willing to wait for that. No clinician, no patient, nobody, no payer for sure.
Yeah, right. Exactly. I think no matter how excited people are about new technologies, I think, the old technology of what's known as turnaround time kind of almost trumps everything. So specifically, where do you have an eye towards your technology being deployed? Do you see it more as being incorporated into clinical diagnostics or as a research tool or a combination?
Well, so I would say, today it's a research use only labeled technology. Our primary customers are biopharma companies that come to us, like they all do, with, very specific need in mind. And if it's in the field of Immune-Oncology, they're typically looking at, what I would call the usual suspects, right? The CD3 that labels the T cells, some of the CD68 that labels the macrophages. But then they're asking, a specific question. So maybe about a specific subtype or a specific target. So what we do for them is we have a process called UView that allows us to make a novel panel for them. But then at the end of that process, if everybody's happy with the panel, they can run that on their machines, or we can actually run it on our machines. We have a CLIA certified lab where we can do that as well. So like we're I would say our activity right now is just mostly driven by the curiosity and desires of the primary customers right now. As you know, it was fortunate to spend several years in drug development in pharma companies and have some understanding of how assays transfer from early stage clinical trials, for example, to later stage clinical trials. And there's several issues, right? There's performance, there's lab environment, there's issues around the protocol and data integrity and statistical analysis plan and all kinds of things. So I think we are working with some customers on exploratory objectives of clinical trials. So we do routinely process clinical samples. But as you know, with exploratory objectives, they're research, right? They're still research. I guess we're excited and I would say, a quality journey to enable eventual use of our technology in clinical contexts. But it is a journey, as you know. It's not something you just, from a standing stop, jump 10 feet and land where you want to be. It's a progressive process. It's a matter of gaining trust, gaining confidence, publishing results, perhaps some degree of standardization within the field around what results mean, using different platforms and different technologies, which of course there are many now, to multiplex.
Yeah, there are many. So I think, one, you're talking about kind of the journey of going from drug discovery to drug approval to actual implementation in clinical practice. And then one of the big developments in diagnostics has been the idea of the companion diagnostic.
Yes.
And I think we've kind of, for whatever reason, as a specialty, or it goes broader than that, we've kind of lagged behind. But the ideal would be to incorporate the companion diagnostic, or what we perceive as the candidate companion diagnostic that will ultimately eventually be used
Right.
to select the patients for therapy, to incorporate that into the clinical trial at the time that it's being done. So it can be done truly in a prospective way.
Right.
So are we there yet to do that?
So I don't think the field is there quite yet. I think if you look at CDXs that are approved, and I just want to clarify, I think that's a common term, but I think it has two meanings out in the field, right? It means it's a concept, right? The idea that you can take a diagnostic and use it to, help decide whether a patient's going to respond or not. The other, I would say, is sort of an official IVD designation. It's a category that is recognized by health authorities as a specific regulatory designation. So I think when you're having discussions about CDX, it's really important to clarify really what you mean. And of course, in the latter, there's also this concept of the complementary diagnostic, which does some of the same things, but not in the same definitional way. When we were talking to folks, we really want to just make sure that they understand exactly what their ambitions are. I would say, right now, we're at the stage where we're using the technology to help our customers, help some academics as well, get a better understanding into simply what they're seeing in tissue. This is often done on retrospective cohorts, large tissue collections where there's already an outcome data available. So this is an excellent opportunity to test hypotheses, to generate hypotheses, potentially just to serve as a basis of real-world evidence. But if you take that to a health authority and show them that, they'll turn you away and say, all right, we'll come back with prospective data, a better lockdown assay. A larger issue is whether the CDX, if we're talking about a designation, is actually, a distributed one or one that is performed in a single site, in essence as an LDT, but as one that's more mature as an LDT, a laboratory developed test. So, I love the concept. I guess I'd like to think historically about this. HER2 is a mature field. The test, as it was designed, over 20 years ago, does a decent job of separating a largely bimodal population of patients into two different categories, right? One that responds pretty well to a targeted therapy and the other ones that don't. And there's 5% in the middle, of course. And I think at that time, and I guess thinking of my 20 years in this field or so, I think the people studying oncology thought that there would just be like a lot more HER2s out there to find, where you'd have these bimodal populations and devise a test, and it would very cleanly separate the responders from the non-responders. That's true of many, mutation-based targets. So, BRAF, KRAS, EGFR, and so on. I think it's probably not true, for example, with PD-L1, right? PD-L1 is introduced and sort of clarified a lot of the potential problems that applying the HER2 paradigm to companion diagnostic development entails. But on the other hand, that's foundational for really the field of Immune-Oncology and testing and sort of transforming cancer care. So, we're in the process of learning a lot still about cancer as we try to help patients with cancer and companies try to develop drugs.
Yeah. Interesting. Yeah, I think, HER2 is kind of the poster child or the paradigmatic case of the targeted therapy and the companion diagnostic. And on the surface of it, it looks pretty good, right? But then when you start poking around, you're like, oh, wait a minute, this isn't what I thought. Going from how the trials were designed, right? Did they truly ever incorporate any HER2 negative patients? So it was basically an enriched trial. So you're starting with all positive patients. And then what's the criteria to enter? Is it 2+? Is it 3+? It wasn't the original assay that was used in the pivotal trial that actually got developed to do a diagnostics. And then you're faced with all kinds of bridging studies. Then you come in later with alternative methodologies such as FISH, and then even in actual clinical practice, HER2 testing was kind of a disaster there in the US, like in the early 2000s.
Right.
Before ESCO and CAP stepped in, so just a lot of issues like that, and then labs were in actual clinical practice, they were tasked with saying, okay, you have to have 95% concordance with a validated method. And no one says, well, wait a minute. The actual concordance with the assay used in the clinical trial was only 79%, right? So it's like there's a lot there, I guess, to learn from and improve on.
Well, and look how long it's taken, right? 2 decades. Yeah. I think the latest CAP update was in 2018, and that really concerned like how you interpret various FISH tests, and not everybody does the same FISH test. It's taken 2 decades, and now, of course, there's new HER2, there's therapies designed for HER2 low. So, the early iterations of the test really didn't require you to distinguish, for example, a 0 from a 1. But now it's going to be important. So the assay itself may have to be looked at again at those cut points. So it's complex business, right?
Right. Yeah, exactly. It's complex and it's evolving, right? Even after all these years, it's still evolving.
Yeah.
So,
which makes it exciting, frankly. It makes it exciting because it's always changing. And you talk to people who practice regularly that, they have to stay on their toes to do the best job they can do and want to do. They have to learn constantly. So it makes it a great career choice, frankly.
Yeah. And it's still kind of bringing this back to kind of what you're working on now. I think the poster child for Immune-Oncology, there have been a few blockbusters launched in the past few years. And then the marker we're looking at is PD-L1, which has somehow become the companion diagnostic. And I think the landscape is different now. I think, when this came out, maybe around 2016 or so, in terms of, our ability to actually do molecular testing and develop markers. And I think a lot of people were kind of scratching their heads and saying, well, why, based on everything we can do, right? Everything we can know, this is the best way, or this is the best companion diagnostic and a single IHC. So I think a lot of people are kind of scrambling behind the scenes and working on alternative methods.
Right.
And you talked about incrementalism, I think, before when we started. And so it kind of sounds like your approach of multiplexing to attack this problem, is more of like a quantum leap, not so much incrementalism if we're talking about multiplexing compared to a single marker. So maybe tell us a little bit about the work you're doing specifically with Immune-Oncology.
Well, all right, just to frame that up, I think before I would call it a quantum leap, I would say it's a quantum leap on the cool scale. But then the question always becomes, how is it useful and how have you shown it's useful? And is it deployable, right? So I don't think you touched on it, but like one of the major complaints about, I think PD-L1 I've seen over, the past several years is just the fact that the assay platform, the clone, they weren't standardized up front. So the foundation of this was really, each different company having a fundamentally different hypothesis about how the drug worked and how they were going to enrich for patients and how they were going to design the assay. And of course, the platform was designed. So, it worked, which is great. Most of them don't. But then now we're left with like, how do you actually test? And then at the same time, what you alluded to it, where it's like, okay, well, all right, we just have a single IHC assay, it enriches, but there's a lot of false positive and false negative responders, right? In whatever assay you use. Heterogeneity confounds testing. So you can do repeat testing and be positive or negative. If you're negative or positive the first time. There's the fact that it varies by indication, right? In some indications, you don't need a test. In others, it probably is really important because it keeps you from getting a drug for which you'll, have no benefit and you might have the opportunity for serious side effects. So, and then, in the meantime, field of immuno-oncology, even though it's a couple of decades old, is still kind of in its infancy from a targeting standpoint. And everybody's getting, combo therapies. So I think when you have a new standard of care, right, it becomes impossible to repeat the trials, impossible and unethical to repeat trials where the standard of care is, a placebo or an old standard of care. I think you're at a point where, you have to begin to, at least in part, rely on some real world evidence, to change practice. And I think the FDA is starting to recognize this. I think this puts a lot of potential power in the hands of individual labs that are innovative and want to offer something new and participate in studies, the right types of studies, and then eventually perhaps take that test to the FDA as a single site PMA. This raises a larger question. If you look at the type of IHC we've done for the last 50 years, right, it's a single slide, run on an autostainer. It's read by a pathologist. There's no software per se. It just has to run well and look good at the end. That can be done globally. right? Because all you need is a microscope. But if you are going to, look at, let's say, 6, 8, 12 markers, you're going to be quantifying phenotypes, you're going to be doing image analysis, is that even scalable? Is it standardizable? I'd like to think it will be one day, but I think we're nowhere near that. So I guess this raises a thought that I've come up with before, right? The FDA has proposed this concept of the pixel pathway as a means to specify whole slide imaging systems. In other words, the ability to track the pixels generated from the scan slide all the way through from the scanner through the software to the viewer. And so that would allow the pathologist to know that when they're looking in the viewer that they're seeing exactly or roughly what they would see in a microscope. I think if we apply that same concept to higher level workflows, more complex workflows, and multiplex is not the only one, I don't think we'll ever standardize it. I think it's going to be too complex because there's too many different ways to do it, and we really don't know the best way. And every system is frankly different. So I guess what I've thought about, since the goal of most of these assays, these multiplex assays that are done, is not just to paint the tissue with 6, 8, or 12 colors. It's actually to identify cell phenotypes and to localize them and then to be able to count them. Why not focus standardization on that attribute? So in other words, let's define an exhausted T cell as this much CD3 relative to the range, this much PD1, this much of other exhaustion marker. What I like about the fact that there's so many multiplexing technologies now is that there's actually the possibility for that data from different modalities to converge on these agreed definitions of cell phenotypes that we can eventually agree on as pathologists to look at in disease, either in a research or eventually in a diagnostic capacity.
I think standardization, is going to be key, especially with all these various competing platforms and methodologies. And I think it gets even more exciting and challenging as we move into digital pathology when we can incorporate H&E features, right?
Yes.
So, maybe the other part of the story for immune-oncology is the good old-fashioned lymphocyte, right? That we've been looking at on H&E sections for a long, long time. And we've known that they have prognostic value. But now we're beginning to enter an era where we can actually quantitate these things in a reliable way using image analysis and then incorporate those into algorithms to help perhaps assess prognosis or who might better respond to therapy.
Yes.
So how do you see that playing out, and maybe even in concert with, say, your multiplexing approach?
So first of all, it's incredibly exciting, right? Just to see the expansion of all the papers where deep learning has been deployed on H&E tissue collections. And, there's several companies now whose goal is really to bring these tools and, to research and then ultimately to the clinical arenas. They're mostly focused on better interpretation of the H&E slide. And of course, this is we have a 150-year legacy in the discipline of knowledge, accumulated through H&E. And this is, of course, how pathologists learn pathology. So almost in parallel, and I saw this in, when I started in industry several years ago, computer vision and image analysis techniques came up, and it was fairly straightforward to take these programs. For example, Visiopharm among them, more recently, Indica Halo is a big player. We use both of these platforms in our company. But it's very straightforward to take, for example, a hematoxylin stain slide, and then it has a light blue stain, and then quantify the brown pixels, because there are some spectral overlap, and it's pretty easy to see dark brown. Even a computer, pretty easy to see dark brown on a light blue background. So you can get positive pixels over a region of interest. You can count cells. You can mimic and count individual cells. You can write algorithms based on purely computational features, nothing to do with machine learning. And you can get numbers that are pretty good. And it's been done in industry for quite a while, right? So AI comes along, and it actually is used by the image analysis companies to help, to learn to segment areas based on learning from a specific sample. But I would say that technology is being applied to its traditional use, in a very different way, in which, several companies have taken large collections of H&E slides and used deep learning to learn new things. So now we have these almost, I would say, two completely sort of parallel lines of investigation going on, image analysis and computational pathology without AI, or with AI used for an image analysis purpose. And then you have deep learning looking at H&E collections. So then we bring a technology that allows you to deeply phenotype each individual cell. And then the question becomes like, what do you do with it? I mean, yes, it's cool, but how's it going to be useful? And I wish I knew more about this, but I have to say, based on what I've learned and read and the people I talk to, is that the field doesn't know. We don't know yet how the best use of this multiplexing is, right? On a very simple level, for a pathologist to be able to access the data, all they need to be able to do is see it, right? They just need to be able to see the sort of the triple positive T cells in the tumor, maybe get a quantification. This doesn't require deep learning, right? It requires formatting data and making it pretty on a screen. So then if you think about what H&E is being used for, with deep learning, what deep learning has the capability of doing, in essence, is potentially seeing things that pathologists can't see or is not trained to see or should be looking for. I mean, I don't know about you, but like when I look at a sea of lymphocytes that are pretty monomorphic, which most non-activated lymphocytes are, either as a lymphoid aggregate or maybe, TILs in a tumor, they all look alike to me. So I don't know. I have no idea how AI might be able to tell that one of those is an exhausted T cell and the other one, is a memory B cell. I don't see it happening, but that's the beautiful thing about science is that I love to be proven wrong. So I believe that this deep phenotyping is going to be incredibly useful for something, certainly to identify the phenotypes of specific cells and infiltrates, to characterize, better characterize hot tumors, to understand checkpoints that are active, maybe identify other types of cells that are preventing your immune response. I know it's going to be useful to identify those cells, but I don't know today how we can use AI to enhance the diagnostic process, right? One possibility is that you merge the H&E, I think, with the MIF, in essence, generate this novel data set that no human has really ever seen, right? It's, there's H&E on top of the self-phenotype characterization, which could be 8, 12, or 16 colors, depending on the combinations you're looking at. Like no human can see that. They can't process it. So you feed that multidimensional data set into, a neural network, and you don't know what's going to come out. It could be incredibly enlightening or it could be confused. I really have no idea. On the other hand, another sort of, I guess I think of this as a simpler way to use it. How do we use NGS right now? The pathologist just picks a good section, right? They send it to the molecular lab. They do all the sequencing. There's no tissue context. But then you have maybe a panel of IHCs where there's tissue context, but no sequencing. It's the pathologist's job to kind of cobble it together in the form of a report and interpret it, right? So you could use MIF data in the same way. You could just say, well, it's more information around staining, and then we'll just let the pathologist integrate it with this other, mutation information, and then that's how you figure it out. So I guess what I'm saying is that I don't think the field knows how to best use this, deploy this technology with any multiplex phenotyping technology, spatial transcriptomics, whatever it is. But we need to do experiments to figure it out.
Yeah. I think we kind of picture, or it's intuitive, we're going to have some kind of future where digital pathology is the platform, and then we incorporate multiplexing based on molecular data or DNA hybridization and so forth, and then incorporate that with H&E features, and then also incorporate that with counting of lymphocytes and classifying the various populations. And somehow we're going to come up with some kind of integrated diagnostic. How realistic that is, when it's going to happen, what it's going to look like. I think there's a lot of unanswered questions.
There may be options, right? Look at Lynch syndrome testing, right, for microsatellite instability, right? There are molecular assays and there are IHC assays. Pathologists like to see things. So if they are asked to establish a condition or diagnosis, I think they would rather have something they can see than something they can't. In part, I think was the motivation over 15 years ago, right, for creation of the Lynch markers and looking at those markers in tissue to make a declaration. Now again, that doesn't give you molecular information, but on the other hand, the MSI PCR tests don't talk to you about heterogeneity per se, right? You can only see that when you do the stain in a tissue. And I think if you look at, testing for NTRK, right, there's three, TRK receptors, ABC, that can be fused with other partners in a variety of tumors. Overall, quite rare, but I don't think there really is a consensus on how you best test for these. You can, in some contexts, IHC works fine. In some contexts, it's confusing. You can do FISH, but then, of course, FISH, you're betting on the translocation, and if it's not the right partner, you won't see it, so it looks normal. Maybe people thinking RNA-seq is the best way to do that, but not everybody has access to RNA-seq. So I think you have to really understand what the lesion is that you're looking for, and then understand really the best way for the patient in light of what you can do to test for it. So, and as you know, with some lesions, there are options.
Yeah, I think you're right. I think we need to, marry what's practical versus, what's out there and come up with solutions that will ultimately benefit patients. So, Keith Wharton, thank you so much for being with us. Before we wrap up, let's learn a little bit about you. Tell us about your early experiences and how you came to be interested in digital pathology.
Oh, wow. Thanks for that question. So in med school, I became really fascinated with molecular biology, how genes work in organisms. And of course, this was in the 80s. Best way to do that then was to look at mutant animals. And so I worked on Drosophila. And so doing that, I did a lot of bright field IHC or immunocytochemistry staining, we'd call it, a lot of microscopy in whole fly embryos. And so when I picked a medical specialty, pathology was a pretty easy choice because you get to spend so much time on the microscope, right? And I'm sure you perhaps made the choice for the same reason. But I trained in the 90s when there wasn't really much of such of a thing as digital pathology. I'd heard from Ron Weinstein his efforts around telepathology, but this was they used cathode ray tubes and things, and so this was early days. But I did take a lot of pictures, both for conferences and as part of my PhD. So I love picture images. Continued in science working on Drosophila, and as a faculty, I managed a research microscopy facility. I did a lot of pathology teaching, so I felt like there was pathology was really sort of focused on its H&E and IHC panel, but then I also got to do this cool confocal microscopy and multiplex labeling. So I was always thinking of ways to kind of marry the two. After running a lab for a bit, so mid-career, I entered industry and I got a job working in a translational pathology lab department, sort of a mid-sized biotech, Biogen at the time. And there, and in a subsequent position at Novartis, I was able to use a lot of image analysis, do a lot of digital pathology, and see how pathology could be applied across the drug development spectrum, from early research through safety assessment and early and late stage clinical trials. And so actually with the founder of Flagship Labs in Colorado, we wrote a book and we helped write and co-edit it around molecular histopathology and tissue biomarkers with a lot of emphasis on image analysis and digital pathology. And it was published now about 6 years ago. So it was a great experience, working with the experts in the field at that time. So anyways, I will finish the story. So after 8 years in pharma, I became more interested in diagnostics as a field. And so I most recently served as a senior medical director at Leica Biosystems. Leica is one of the major scanner manufacturers as well as reagent and staining and really the whole histopathology workflow. So I supported the whole business, which was really exciting. And I was fortunate to be a part of the Aperio team that was able to obtain the second FDA clearance for pulse light imaging system. This was the AT2DX in digital pathology in 2019. It was really kind of a landmark for the field after the Philips Telesite approval a couple of years earlier. And I also worked closely with the DPA, Digital Pathology Association, during that time. So incredible privilege because, of course, you meet a lot of different stakeholders in the digital pathology world. So I've been at Ultivue for the last 6 months as a medical director. And so, as I mentioned, we're totally dependent on digital pathology, in our shop. We work with a lot of pharma clients, as I mentioned, on really cool and we think important multiplex projects, and also with a variety of stakeholders to understand how these technologies might be used eventually in a diagnostic context. And I'm really, keen to understand what that road is going to be like, because I think it will be, I guess, the way I think of it is that the pathologists already do some multiplexing, right? There's PIN4, there's a few other panels they use for very specific diagnostic decisions. I think what multiplex does allow is it allows you conceptually to do something you just can't do with single IHCs. You just can't do it. So to identify, for example, marker triple positive or quadruple positive cells, and to be able to precisely quantify them in the H&E. So yeah, that's where I am now. It's an incredible journey. I think we're just this exciting stage where there's a lot of technologies out there. There's a lot of information coming from the single cell phenotyping studies that are going to be informing new panels and helping us identify these novel cell types in disease.
Yeah, this is a very exciting road to be on indeed. So just before we wrap up, what excites you? Where do you see things going in the next 10 years or so?
Yeah, so in the next 10 years, I think about many of the topics we touched upon, but a bit of a convergence so that these modalities are all brought to bear in the environment of a pathologist. So I think about, and I remember seeing Dirk Sunsken, the original founder of Aperio, talk about this, introduce this concept at meetings 15 years ago or so, the concept of the pathologist cockpit, right? So bringing everything together on the screens in front of a pathologist to let them navigate between, the data and the diagnosis. So having that cockpit come together, having that be an intelligent cockpit, having it be one that's visually striking, that they can trust, having it be one that's ergonomically friendly in a way that, for example, electronic medical records are typically not friendly and have been attributed to contributing to burnout of physicians because of all the time they spend filling out the EMR, the so-called death by 1,000 clicks. We don't want the experience of the pathologist to be like that. We want it to be something that enhances their experience as a pathologist and makes them more effective. It's really a convergence, I think, of all of these things coming together and just, it being better for everybody.
Yeah, that's great. The great convergence and making us more effective and better for everybody. Well, our guest has been Dr. Keith Wharton from Ultivue. We'll see you next time on Digital Pathology Today.
Thank you.
Thank you.
This has been Digital Pathology Today. Please be sure to subscribe. Thanks for listening.